This passage is adapted from Paul South. Helping plants remove natural toxins could boost crop yields by 47%. When it comes to photosynthesis, plants use sunlight to power a chemical reaction that converts carbon dioxide and water to sugars and oxygen. But that isn't the only chemical reaction that occurs in plants. A quirk in the evolution of the protein called rubisco is that sometimes instead of converting carbon dioxide during photosynthesis, it uses oxygen instead. This produces waste products such as glycolate and ammonia, which can be toxic to plants and slow or stunt their growth. To remove these toxic chemicals, another process needs to kick into gear. Photorespiration is a part of natural plant metabolism that recycles these toxins. It is a necessary process in major crops, including rice, wheat, and soybean, as well as most fruit and vegetable crops. Recycling these toxic byproducts sucks up a huge portion of the plant's energy and can inhibit the plant's growth by more than 30%. At higher temperatures, plants tend to increase the amount of oxygen they convert. So, as the growing season temperatures rise and heat waves strike, up to 50% of the energy generated from photosynthesis can be required for photorespiration to recycle toxins in major crops like wheat and soybean. That slashes yields in the hotter and drier regions of the world, such as sub-Saharan Africa and Southeast Asia, where food is most needed. To meet the growing demand for increased food production, I worked with an international team to explore whether speeding up photorespiration might boost crop yields. The work, led by Professor Christine Rains and Patricia Lopez Calgagno, explored whether this modification could boost the production of tobacco plants. We managed to speed up the recycling of these toxins by designing plants that produce more of a protein called the H protein that is already present in our crop plants and plays a role in photorespiration. Previous work in the lab using the small plant Arabidopsis, the lab rat of plant research, suggested that increasing the quantity of H protein could speed up photorespiration and enable our plants to grow larger. Our team translated this idea from the lab to the field using a strain of tobacco, Nicotina tabacum, which we grew outside at a research field station. We discovered pretty quickly that we had to carefully control the quantity of the H protein we engineered plants to produce. Too much H protein in all parts of the plant was harmful, stunting growth and reducing yield of tobacco leaves. Thus, we fine-tuned our approach in engineered plants and manufactured the H protein only in the leaves. This increased photosynthesis and plant growth, probably because of faster recycling of the toxic chemicals. We tested our hypothesis in tobacco because it is an excellent model for proof of concept research. It is easy to genetically engineer and only has a 4-month life cycle, allowing us to conduct several trials in one field season. This allows us to test various genetic modifications in tobacco and then translate those discoveries to make improvements in targeted food crops. To fine-tune the expression of the H protein, the team engineered the tobacco using DNA from a close relative, Solanum tuberosum or potato. Using a known sequence of potato DNA, we were able to boost the H protein specifically in the desired leaf tissue. That proved to be the key to increasing yield without harming the plant. Initially, I was skeptical that boosting the production of a single protein out of thousands in the plant could have such a dramatic impact on crop yield, but after 2 years of field trials, my colleagues and I have demonstrated that increasing H protein levels leads to larger plants, boosting crop yields by 27 to 47%. Which choice provides the best evidence for, 42 best evidence. No, I think this question had to say something else. Let me check. May SAT. Yeah. Question 42. Over the course of the passage, the main focus shifts from a discussion of. Okay, that's the question. So it's not this. over the course of the passage, the focus shifts from. Okay. Plants' use of sunlight and photosynthesis to an analysis of previous attempts by researchers to prevent oxygen conversion during photorespiration. Naturally occurring chemical processes in plants to an account of experimental efforts to examine the effects of certain genetic modifications on crop yield. Impact of climate on crop yield to an evaluation of a hypothesis about how temperature affects production of a particular protein in plants. Proteins essential to the removal of toxins in plants to an explanation of how those proteins important to photosynthesis was first identified in a laboratory. So here the answer is B. We are talking about these naturally occurring chemical processes, right? We start with photosynthesis and then we explain what photorespiration is and how it removes toxins. But then the problems with photorespiration in terms of energy used by plants. And then it moves to experimental efforts to examine how you could have certain genetic modifications that can enhance crop yield, right? Option A, use of sunlight to an analysis of previous attempt. No, the focus is on current experiments. Impact of climate is very briefly touched upon in terms of energy yield. And yeah, and this is not talking about the current experiment. As used in line 2, power most nearly means to power a chemical reaction. They use sunlight to enable a chemical reaction. Activate, right? Activate is the closest to enable in this context. Authorize, engineer, or dominate. They don't fit the context of a chemical reaction, right? Enabling a chemical reaction. Which statement about the production of toxins resulting from Rubesco's conversion of oxygen in certain crops is best supported by the passage? Oxygen conversion results in higher levels of toxins in major crop plants than it does in other plant varieties. Presence of toxins results in a greater reduction in plant growth than does photorespiration. Environmental temperatures decrease, photorespiration recycles the toxins in the plants less effectively. As environmental temperatures decrease, the amount of toxins decrease. Is this evidence-based? Yeah. So I think I'll go with D, because here it says that as growing season temperatures rise, up to 50% of the energy can be required for photorespiration to recycle toxins, right? So it's about efficiency. Amount of toxins decreases. No, it's not about the amount. As environmental temperatures decrease. Is this the correct option? I'm just going to confirm from the other one. Yeah, it is. As environmental temperatures increase, right? Photorespiration recycles the toxins of the plants less effectively. Is it? Yeah, so it is. Yeah, so it's not C either. The presence of toxins results in a greater reduction in plant growth. Let me just work with the line numbers. Let's mark the line numbers and then we'll be able to figure this out. 1 second. Yeah. Okay, 14, 16, 17, 19. 14, 16, 17 to 19. And 19, 25, 25, 28. 19 to 25 and 25 to 28. Okay, so it is necessary process as well as most fruits and vegetables crops. So it's recycling these toxic byproducts sucks up a huge amount of plants' energy and can inhibit the plant's growth, right? So that does have an effect, right? What was the point saying? Presence of toxins results in a greater reduction in plant growth than does, yeah, the presence of toxins is the main cause of the reduction in plant growth. Photorespiration by itself is not a problem. It's a necessary process, but recycling these toxins sucks up a huge portion of the plant's energy, right? Right. So the presence of, so I like B, oxygen conversion. Yeah, there's no comparison with others. So B is a good option. And yeah, and B for this one as well. When the author says that he and the other researchers translated this idea from the lab to the field, he most likely means that they applied a concept to a new environment, predicted, where is he saying that? 45 to 46. Okay. Our team translated this idea from the lab. Okay, so they had a previous work had been done in the lab and then they translated it. So they applied it to a new environment. I like this. Predicted variable study results based on a new setting, adjusted their hypothesis to account for a different location, changed the experimental process. No, so it's these verbs don't make sense in the context of that statement. It's just applying the concept to a new environment. The passage indicates that the use of solanum tuberosum allowed the researchers to reduce the amount of, is this evidence? No. Glycolate and ammonia produced in the tobacco plants, localize increased production of H protein, confirm whether boosting production of protein other than H protein. Yeah, I think it's B, right? Because they were able to localize it in the leaf. Yeah, using a known sequence, we were able to boost it specifically in the desired leaf tissue, and that came from the close relative potato, right? So it is localizing increased production of the protein to an area that is most beneficial. The main purpose of the last paragraph is to, what is the last paragraph saying? Initially, I was skeptical, but we did get the desired results after 2 years of trials, right? So, is to express a researcher's sustained surprise about a finding, illustrate the evolution of a researcher's confidence in a hypothesis, highlight the time-consuming nature, endorse the simplification of a technique. So it's B. It took him time, right? He was not initially sure about it, but over time he was certain that this worked, right? It's not sustained surprise, it's just an evolution of his confidence. The time-consuming nature is not what he's highlighting, and the technique was not simplified. Okay. Information from the passage best supports which potential explanation for the data in figure 1 regarding the leaf area of H protein increased strains. Okay, figure 1. Seedling leaf area 26 days after sowing in control and H protein increased stains. H protein increased stains. Okay. So I'll just open this on another page as well. Okay. So we are trying to find a reason for, okay, Arabidopsis DNA to produce more H proteins. Okay, there's something wrong with this option. I'll just confirm it. The G, okay, G20 and G. So this part has to come first. The G20 and G46 strains were modified with Arabidopsis DNA to produce more H proteins without harming the plants. Okay, the C, G11 and G21 strains in amounts that damage them. Okay, photorespiration occurred at a faster rate in these, and H proteins were more concentrated in the levels of this than they were in this. Okay, seedling leaf area. Okay, I'm just going to mark the line numbers because that will give me a sense of what we are working with. Uh, 1 second, I'll just mark this. Yeah, let me change the color. Okay, so we have 41 to 45, 41 to 45, okay. And then we have 49 to 51, 49 to 51, okay. 51, 53, 55, 57. 51 to 53 and 55 to 57. Okay. So previous work in the lab using the plant Arabidopsis suggested that increasing the quantity of H protein could speed up photorespiration and enable the plants to grow larger. We discovered pretty quickly that we had to carefully control the quantity of H protein we engineered. Too much H protein in all parts of the plant was harmful, stunting growth and reducing yield of tobacco leaves. Okay. Tobacco leaf yield was reduced. That is what we are interested in, that diagram. This increased photosynthesis and plant growth probably because of faster. Yeah, so I'm working with C now because I'm working with C because that diagram, if you look at that diagram, it is interested in the yield, right? The leaf area, which was, yeah, so the yield was reduced. So, right, so now photorespiration occurred at a faster rate. Yeah, so the CG11 and they produce H proteins throughout the plant in amounts that damaged them. That is why the yield was less. Okay, based on figure 2, the researchers' most successful attempt to boost tobacco yield resulted in a mean plant dry weight that was approximately what percentage of the dry weight of control plants? Okay, so let's look at figure 2. The researchers' most successful, so mean plant dry weight, percentage of dry weight, okay, mean plant dry weight was most resulted in a mean that was approximately most successful attempt. Okay, trial 1 and trial 2. Tobacco strains. Okay, control. So we are talking about, yeah, growth, right? So it was most successful here. G46. So that's a little over 140. Yeah, so 145%. Based on the passage and figure 2, which strain of H protein increased tobacco plant has data that are the furthest from the results presented in uh, presented in lines, okay, 76 to 79. Furthest, right? So let's look at 76 to 79. After 2 years, my colleagues and I have demonstrated that increasing H protein levels leads to larger plants. So which are the furthest from that? Okay, let's look at the figure now. So G20, because G20, the dry weight was much, much lower than the control. So G20 in trial 2. Yeah. Okay, let's grade this. 42. $42. BADCA, BADCA. Okay, so this is D and this is C, BADCA. Okay, what's the next one? 47 triple B triple C. 47 uh, 47 B B B C C C.